Hi, my name's Taser Ray, a research scientist with our company in Craft We Trust. I'll be presenting breakthrough tech today on how to grow in one cycle pest-resistant cultivars, something you've likely never seen before. So let's get on with it. There's a lot of information to cover today.
Yep, lots to cover today. First, a big thanks to the Growing Summit for the gathering of your green minds, plus receiving the privilege of presenting our breakthrough technology, cracking the genetic code using friendly fungi. Where did all this start? Well, I've been a researcher for over 36 years, starting with chasing caribou and grizzly bears around extremely mountainous terrain. And wow, there's some crazy stories there. And then I went on to environmental monitoring, studying the effects of living in ecosystems from mining, forestry, industrial manufacturing, studying the effects at the macro and micro levels. For the past 11 years, I've been learning more intensely how to work with our communities of organisms at the cellular level. These organisms naturally reside in plants, animals, and people, the ones we don't normally see. The cutting edge tech I've learned to do is communicate with friendly fungi, the ones that live in harmony with plants, animals, and people. What we're covering today is how these friendly fungi receive and act on instructions given using our space age tech, a biocommunicator tool. How did I come up with this biocommunicator tool? Short history here is necessary. I moved to Hawaii about 11 years ago. While there, I was used to use my kind of like fringe science knowledge to come up with a new type of mosquito repellent. I looked into it. I found research articles from England documenting how the human body has a natural ability to produce invisible vapors that hide it from blood-seeking mosquitoes. Pretty cool. Researching further, I stumbled upon an ancient technology that could allow communication to the micro world of cells. I first thought this tech was even too wild for me, but I was encouraged to try it out. So I modified this ancient tech so I could communicate with the organisms at the cellular level, stabilized in a natural non-toxic mineral water, sort of like a biomessenger program. Now, this ancient technology showed how to record info to be read by the microorganisms. Yep, we're going down the rabbit hole here. Basically, I created an ingestible mineral water biocommunicator tool that communicates at the cellular level. Within minutes of taking this biocommunicator tool, a human experiences the ability to walk through clouds of mosquitoes, black flies, horseflies, and no-see-ums and not get bit. Really, you have to experience it. It's really so kind of a miracle. Anyway, this morphed into an agriculture product. While still in Hawaii, several coffee farmers in Hawaii that knew of my mosquito solution asked jokingly if I could do something for protecting their coffee trees that were being decimated by the coffee berry borer beetle. I said, I don't know. I'll look into it. I did look into it. Here's what I found. The CBBB beetle is looking for two alcohols in the coffee cherry to initiate her reproductive cycle. So I thought, what if I could get the fungi groups in the coffee tree to stop the female beetle from having access to these two alcohols? Seems reasonable. The biocommunicator now morphed for plant use. We find life at the micro level is really fairly universal. Some science articles show me what the coffee tree has for natural protection programs. Plus, the chink in the beetle's armor is those two alcohols from the coffee cherry. The female beetle needs them to reproduce herself. So I instructed the coffee tree fungi coordinators to initiate a program to stop the alcohols being used by the beetle and use the alcohols elsewhere in the tree. After the farmers applied the biocommunicator tool to their coffee trees, their infestations of beetle went down to virtually zero within a few weeks. The farmers also commented to me that their crop production had increased along with the taste and the value of those coffee beans. So in 2015, I'm back in Canada. Now, a few cannabis growers that heard of my success with the mineral waters in Hawaii, they approached me and asked if I could do something for spider mites. I said, I don't know, but I'll look into it. And I did. It's also gave us some really sage advice from a long-time cannabis grower. He said, I found there's two kinds of cannabis growers. One type, they're dealing with a nightmare. Two, they're about to deal with a nightmare. Well, which one are you? Anyway, so with that, I was inspired to find a solution. I found a couple of studies out of Europe that confirmed the cannabis plant has natural pest protection defenses. Two entopathogenic fungi that naturally live in the cannabis plant. These two fungi are scientifically recognized to release spores and to attack pests. So again, I used the ancient technology to communicate instructions to cannabis plant resident fungi groups, directing the fungi to switch on dormant programs in the DNA for pest control. The biocommunicator tool, mineral water, is applied to the plant as a light foliar and substrate drench. Within 48 hours of application, you can start seeing changes occurring. Here's what happens. The fungi respond to the messaging, release activated spores throughout the fluid and plant tissues. The defense mechanisms become activated. Any pest such as spider mite or thrip that comes along and bites on the tissues of the plant gets eaten. Activated spores of protection are ingested into the gut of the pest. These spores rapidly grow, expand from the gut and into the body of the pest. fungus and the pest then transfers minerals back to the plant, sucking the pest dry. Pest drops off dead. Reproductive cycle of pest, nada. Making your IPM program way easier. As a point of interest, the biocommunicator tool used on the cannabis plants also increased the production of flower, THC, CBD, and intensified the terpene expression. We also saw the plants eat about 40% more nutrients than usual because they're getting a whole lot more light and they're just a lot stronger. We documented these results and can supply you with this documentation. We are presently working with a cannabis nursery in BC, growing mother plants that are pest resistant, and in doing so, their babies, the clones, will also be pest resistant. We're looking for more cannabis nurseries to work with that would like to produce pest resistant clones. Now, on to introduce Ryan Murray, our breeder, master grower, consultant. All right. Now I'd like to introduce Ryan Murray. He's been a cannabis farmer for over 20 years, 7-time Cannabis Cup Award winner, all-around nice guy. The CEO of a LP in Edmonton introduced us to their master grower. That happened to be Ryan. We developed a closer business relationship with Ryan as he used our technology. That morphed into him being our consultant. Ryan has been using the communicator tool for over a year and a half now in all his grows and is pleased to share his experiences with using this amazing breakthrough technology. Okay, Ryan, so using this communicator tool on your cannabis plants, give us a short version of the differences you've experienced with typical pest challenges there in Edmonton.
Well, yeah, as Cesar says, I'm up here in Edmonton, and first thing I learned when I got here was that Edmonton is pretty much the thrip capital of Canada. There's currently over 36 species of thrips, and as an indoor gardener, you're bound to get them.
Mm-hmm.
When I started using this product, or the communicator, I had an infestation of thrips. It was really bad. My plants were starting to get weak, and the problem was growing. Within about 6 weeks of using the communicator, the thrips had disappeared. The plants had come back to a very healthy stage, and in a year and a half of using the communicator, I've had 0 bug issues.
Wow.
Right from thrips to spider mites, I've had nothing. So
That's
it's
amazing.
been awesome.
All right, so Ryan, one of the trials you did with the mineral complex was to apply it to some mother plants. Let's hear your story and what happened with that.
Well, when I did the side-by-side comparison, we ended up using that inoculated plant as a mother plant. So I did a clone run off of 2 clones off of the mother, and 2 clones off the non-inoculated plant. The inoculated mother increased. My clones were in soil in about 8 days. The roots there was twice as many roots. The stem structure of the plant was generally stronger and healthier, and the growth behind the inoculated clone happened probably 7 days sooner than the other clone. So in a 14-day clone, it was already almost twice the size as the other.
Wow, okay. So then going on to what other kind of cool things have you seen while using this product?
Well, I think that one of the most apparent things was the size of family. In the initial controlled experiment, the fan leaves on the inoculated plant were almost twice as big.
Wow.
Allowing them to take in more light, take in more nutrients, take in more energy, convert that to the growth in the plant. And yeah, that was one of the biggest, I guess, directly related to that. With the bigger fan leaves, created a stronger structure. The branches were stronger. They almost took on like an I-beam shape, where it allowed them to actually hold the weight of the flower without showing any stress. Those are 2 of the most apparent things. One of the things that we noticed was the increase in food. I probably had to add about 30 to 40% more food on the inoculated plant because it was using it more effectively. It was growing faster, and it generally just needed it. So that was a bonus. And I guess the biggest thing that we noticed is in the lab results, the side-by-side trial run, the initial pheno showed about 7 to 8% THC. The inoculated plant, being the exact same pheno, increased to about 8 or 9% THC and about the same in CBD. And I think one of the most interesting aspects is the fact that the clones that came off of the inoculated plant, its profile continued to increase. So the clone that came off inoculated showed a analysis of over 11% THC and just under 11% CBD.
Wow.
So that was a really cool results to see from the lab.
Okay, well, let's carry on now to get some questions from people and see what they have to say. Hey, Ryan, let's get ready for that.
Sounds good, guys.
Okay.